Bismillahirrahmanirrahim. In this lesson, I will discuss about development and structure of seed. We know that after fertilization in female gametophyte, a zygote divides repeatedly by mitosis and develops into an embryo. At this stage, in gymnosperms and angiosperms, ovule changes into seed and the formation of seed completes the process of sexual reproduction in seed plants. The evolution of seed has been proved as an important step in the success and spread of flowering plants as compared to the seedless plants like mosses and ferns. Angiosperm seeds consists of three distinct parts. First one is embryo formed from zygote. Second one is endosperm and endosperm tissue formed from endosperm nucleus, and third one is the seed coat which develops from the wall of ovule, that is integument. Seed coat or testa develops from the integument, originally surrounding the ovule. It may be a paper thin layer, for example peanut, or thick and hard, for example coconut. This seed coat protects embryo from mechanical injury and from drying out. There is a scar on seed coat, that is hilum. It is where the seed is attached to the ovary wall or fruit. At one end of hilum there is a micropyle. This is the same opening through which pollen tube entered ovule. Seed uses it for the absorption of water. The embryo is actually an immature plant. It consists of a radicle and a plumule and one or two cotyledons, that are seed leaves. The radicle of embryo develops into new root while the plumule develops into new shoot. The embryonic stem above the point of attachment of cotyledon is called apicotyl. So the embryonic stem below the point of attachment is hypocotyl. So here is a huge difference between apicotyl and hypocotyl. Within the seed there is a store of nutrients for the seedling that will grow from embryo. In angiosperms, the stored food is derived from the endosperm tissue. This tissue is rich in oil or starch and protein. In many seeds, the food of endosperm is absorbed and stored by cotyledons. The form of stored nutrients in seeds varies depending on kind of plant. Next, I will discuss about germination of seed. For the germination of seeds, they must arrive at a suitable location and be there at a time that is favorable for germination and growth. For example, if a seed is landing at a cement wall, then it will not grow. So the soil is needed for the growth of this seed. Seed germination is a process by which seed embryo develops into a seedling. So here you are watching on your screen that here is seed germination, and there are different kinds of seedlings. During germination, embryo soaks up water which causes it to swell, splitting the seed coat. In the first step, the seed coat will split. Root is the first structure that emerges from this radicle present in seed. It grows rapidly and absorbs water and nutrients from the soil. In next phase, plumule develops into tiny shoot which elongates and come out of soil. So you are watching on your screen that first there was a seed, then there was splitting, then radicle emerges, then plumule emerges. You might have seen that there are different kind of seeds which are at your home, but they are not germinating because they are in the stage of dormancy. At home you are eating different kind of nuts including almonds, walnuts, and there are different kinds of pulses which have seeds and they are in dormant stage. So most seeds go through a period during which there is no growth. This period is called the dormancy of the seed. Dormant seeds are ripe seeds but do not germinate. Under favorable conditions, the seed break dormancy and begin to germinate. Now I will discuss about apigeal germination and hypogeal germination. Api mean upper and hypo mean down. So the germination which is above the ground is known as apigeal germination. So on the basis of elongation of hypocotyl and epicotyl, there are two types of germination. In apigeal germination, that is the first one, the hypocotyl elongates and form a hook pulling the cotyledons above the ground. So here you are watching, at home you might have seen the seeds of beans. In those seeds you are able to observe the apigeal germination. And in apigeal germination, the hypocotyl elongates and forms a hook pulling the cotyledons above ground. For example, beans, cotton and papaya are the examples of seeds that germinate in this way. And this is the second one, that is hypogeal germination. Hypo means down. So the apicotyl elongates and forms the hook. In this type of germination, the cotyledons stay underground. So you are watching on your screen that apigeal and hypogeal germination are totally different. Pea, maize and coconut germinate this way. The next one is conditions for seed germination. Germination of seeds of many plants is also favored by light. In others, germination is retarded by light. Seed germination depends on both internal and external conditions. The internal conditions include a live embryo and sufficient food storage. The most important external conditions include water, oxygen and favorable temperatures. Water or moisture is needed for seed germination. Seeds of most plants have low water content and germination cannot occur until seed coat or other tissues have taken in water. The absorbed water is used in the digestion of stored food and it also helps in the elongation of hypocotyl and epicotyl. Oxygen is essential for the respiration in the cells of embryos. Last one is temperature. Seeds differ greatly in their temperature requirements for germination. The optimum temperature for the germination of the seeds of most plants ranges from 25 to 30 degrees Celsius. So that was all about germination, apigeal germination and hypogeal germination. I will discuss about sexual reproduction in animals in my next video lesson. Thank you and have a nice day.